Dear Organizing Committee, ladies and gentlemen, good morning, good afternoon, and good evening. I'm very happy to have the opportunity to present recent works. A chromosome-scale genome assemble, a diploid oat, Avena longiglumis defined, repeat gene, and chromosome evolution. My name is Qing Liu, came from South China Botanic Garden. Our group has made substantial progress towards understanding the genomic organization at the DNA sequence level of oat and diploid relatives. Avena includes about 29 species, exhibiting considerable morphological and ecological diversity in the Mediterranean basin, Eastern Africa, Europe, Asia, and the Americas. Hexaploid oat is a recognized health food due to oat beta-glucan, which helps reduce blood cholesterol level and heart disease risk. Figure A to F show A genome diploid species, for example, Avena longiglumis with a solitary long spiculate on a panicle, glum's structure tough and equal length. G to H show C genome diploid species, like figure G show Avena areata characterized by unequal length of glums. Two steps were inferred for oat formation. The first step includes the hybridization between the ancient A and C genome diploid ancestors to form AC genome tetraploids. The second step includes the hybridization between AC genome tetraploids and the more recent D genome diploid progenitors to form the extant ACD genome hexaploid. D subgenome might be the varied A subgenome. Result is consistent with those reported using chloroplast genome data. Based on the phylogenetic chloroplast genome sequences, the phylogenetic relationships between oat and its wild-related species were investigated. The basal position of ALO within clade 1 was strongly supported. Candidate A genome ancestors for hexaploid oat species. ALO is short for Avena longiglumis. A higher quality ALO genome assembled by integration of Illumina, ONT, and Hi-C technologies. The ONT raw date and the Illumina date were used for interactive error correction 4 times, yielding a polished assemble. Further Hi-C date was used to improve the assemble and anchor scaffolds into superscaffold, which were clustered and ordered into 7 pseudochromosomes. ALO genome size was estimated to be 4.6 gigabase by flow cytometry and 3.97 gigabase calculated by k-mer analysis. The chromosome-scale genome assemble size was 3.85 gigabase, occupied 97% of the k-mer estimation result. ALO has a genome size about 10 times larger than unseated glaucoma, having nearly the same number of genes. The repetitive component of the genome is responsible for much of the genome size difference. With our nearly end-to-end assemble, we see how the gene figure circle F are distributed widely along the chromosomes. Transposable elements is abbreviated as TES, which are important contributors to ALO genome structure and evolution. EDTA package is composed of 8 polished programs. It identifies the whole genome TE annotation and evaluation. This pipeline was used to predict TE library, occupying 84% of ALO genome. A total of 3.45 gigabase, representing 87% of the ALO genome could be classified as repeats. We note that ALO assemble showed relatively uniform expansion of repeat sequences along chromosomes. 45S RNAs representing 0.3% of ALO genome located on chromosomes 1 and 7. 5S RNA representing 0.005% of ALO genome on the chromosome 7. Comparison with our fish result, A tandem repeat ABT105, shared widely in Avena, was located at the chromosome centromeres. Compared with other 6 classes, the repetitive DNAs are responsible for the interspecific genome size difference. Allo had a lower proportion of DNA transposons. 10 most abundant TE families represent about 60% of allo genome assemble. Recent bursts of both diploid and copia around 1 million years ago contributed to the allo genome expansion. Gene structure prediction depended on 4 methods: RNA-seq assigned prediction, an initial prediction, homology-based prediction, and masking repeat methods for single-copy gene pool design or oligofish experiment. Total 40,000 and 845 protein coding genes were identified in the ALO genome, compared with other published Pooideae genomes. The gene numbers in ALO is similar but slightly greater than those in Brachypodium distachyon, Oryza sativa, Sorghum bacala, Setaria italica, and Z. maize. With our new ALO genome assemble, we see how the genes are distributed widely along the chromosome. Gene distribution along chromosome is interspersed with multiple families of relatively young LTR elements, which have amplified in the last couple of million years. We clustered annotated gene into gene families among LO and 9 other grass species. A total of 1,885 single-copy genes were identified among 11 species, which were used for phylogenetic reconstruction. In the subfamily Pooideae, Avena diverged from Agelopsis toschi in 20 million years ago. The C genome diploid Avena areata diverged from A genome diploid species around 9 million years ago. A total of 2,277 gene families were unique to Avena species, emphasizing the novel gene pool in Avena. To determine the chromosome structure in Avena, we performed an intragenomic syntenic analysis. Examples of shared major blocks of parallel genes between chromosomes 1, 2, 3 are Avena longiglumis. The block covers most of gene-rich pairs of the genome with no indication of major deletions following whole genome duplication, abbreviated as the WGD. The pairs of syntenic blocks are likely to identify the signature of the ancient row WGD event in grasses. There were no regions with 3 or more copies, suggesting no major segmental duplications. In contrast to Musa and Unseed that do not share the grass row WGD event, there is clear evidence for 2 rounds of WGD in Avena. In order to understand the nature of chromosomal rearrangements across Pooideae, conserved genes for each ancient grass karyotype chromosome have been identified for allo and 6 grass species. Chromosome rearrangement has been restricted to a small number of events, presumably avoiding alteration of interactions of gene groups and promoters along chromosomes without chromosome fusion or fusions. Given the high synteny observed in BOP clade, including Bambusoidea, Orthologous, and Pooideae subfamily, conserved nucleotide sequences and domains can be identified by major linkage blocks in grasses. We suggest that this can form the basis of synthetic pan-chromosomal oligonucleotide pools for fish to identify the karyotype rearrangements across the polys. Conclusion: we assembled the chromosome-scale Avena longiglumis genome length 3.85 gigabase Repeats are responsible for much of the difference in genome size. With our new ALO genome sample, we see how the genes are distributed widely along the chromosomes, interspersed with multiple young repeat families, which have amplified in the last couple of million years ago. By syntenic comparison, we could identify rather limited number of chromosome rearrangement events occurred within the BOP clade, including about half the grass species. A remarkable feature of the Avena genus is the translocation of large distal segments of chromosomes in both diploid and polyploid species. Three future directions, including beta-glucan synthesis gene family evolution, individual chromosome identification by oligofish for Avena longiglumis and BOP clade grasses. We thank National Science Foundation of China, Guangdong Province Basic and Applied Basic Research Foundation, Overseas Distinguished Scholar Project, and Foreign Expert Project from Science and Technology Department of China. We also thank Agriculture Agri-Food Canada and USDA Seed Bank for seed collection. PET has allowed Harrison Lab members Wei Wang, Mingzhi Li, and Hong Yuan for data analysis. We thank Zhong Yisun, Xu Kaitan for the statistic and advice. We thank Grandomics, bioscience company for sequencing support, Huawei Elastic Cloud Server for supplying the large repeat data analysis resources. Partial software used in the study listed here, website for ALO sequencing data, and the genome assemble. Thanks for your comments. Thanks so much for your time.